Let's go. All right. Part 3, let's get crazy. Let's go pyrosequencing. And this is pretty nuts, but it's a really cool technique. And this is on the cheaper end of what we call shotgun sequencing. As when firing a shotgun, the idea is that you have no idea where each of the pellets go. You just ensure that you're going to hit something. Yeah. And it's kind of the same way with shotgun sequencing. You can take a little sample of dog dirt off your shoe and you can sequence the DNA in there and you can find out which bacteria and how many of them there are because all the information is there. The nice thing about pyrosequencing is that you can get potentially millions of sequences done in an hour or two. Whereas each Sanger sequence, well, you start by sending it off in the mail, but it's about the same length of process, but you only get one sequence at the end. With a pyrosequencing run, you get all the sequences. You can sequence a genome in a couple hours. Yeah, which is pretty nuts, but why not? Okay, this is basically everything that's going on. I think this is going to make no sense right now, but when you come back to this at the end, it's going to make perfect sense. I'll explain it anyway, and then we'll go through it and actually explain it, because, yeah, you deserve that. Yeah. Okay, so we're going to do PCR, but instead of doing PCR in a little tube like people do, yeah, we're going to do PCR attached to a little bead, and I think it's even a styrofoam bead if I remember right, polystyrene. And so we're going to do PCR on this bead. That's weird, yeah. We're going to do millions of those PCRs at the same time. Like, that's crazy. And we'll do them all in the same tube, and they won't get mixed up. Each bead will have the sequence of one individual piece of DNA all over it by the end of it. That's pretty cool, right? Yeah, that's some next level stuff. When you see how it's done, though, it's like learning the secret behind a magic trick. The only response is just, oh. Oh, okay, yeah. I get it, the magic's gone. And that's the feeling I'd like you to have by the end of this. Yeah. So then, once you do this, then we're going to go in and we're going to do, basically, it's like PCR or Sanger sequencing, except without the dideoxys. So we're going to have one primer and we're just going to sequence out, and we're not going to stop, because why would we ever stop? And we're going to, every time we put in a nucleotide, we're going to release a couple phosphates, and because you start out with a triphosphate, right? And you put it in, you lose two phosphates, and you have one phosphate left that joins to the previous nucleotide. That double phosphate, which is called a pyrophosphate, which is totally the same reason why it's called pyrosequencing. Really, it's pyrophosphate-based sequencing, but to its friends, we can call it this. So we're going to take that, we're going to turn it into an ATP, not by magic, but by a handy chemical reaction. We're going to take that ATP and we're going to feed it to another enzyme called luciferin. That's the one that makes fireflies or lightning bugs, whatever you call them, glow at night. Yeah, if you're from a place where that happens. And so we're going to do this reaction, and that's going to make a little yellowish green light, really cool looking. And then we're going to have a camera, and we're going to take a picture, and it's going to look like this. And so we're going to have millions of little wells, each of which is sequencing DNA. And we're going to look and see, are the lights on or are the lights off? And how long are the lights on for? And that's the whole readout. And by now you're saying, no, that doesn't work. But it does. It does. Yeah. Watch. You'll see. And then it's like a cleanup stage, but whatever. Cleanups for losers and for about 3 slides from now. So let's go. All right. Step 1, you got to make yourself a library. Yeah. And by library, we just mean we've got to prepare the DNA to get it ready for sequencing. So step 1, DNA tends to come in rather long chunks. I don't know if you've looked at your chromosomes lately, but they're pretty big, like billions of nucleotides big, I think. Yeah. So we need to fragment it, because these techniques can only read little bits of DNA accurately. And depending on which version of the technique you're using, and there are a bunch of versions, I'm covering one here, the sequence that you can read is like 50 to 250. But the cool thing is, you can read 50 to 250 from either end of a molecule. So you may get, 250 here that overlaps with 250 there, and so you may get out like, I don't know, almost 500 nucleotides, minus whatever the overlap is from this. Or you might get this end of the sequence and this end, which is over here, and you don't know what's in between, but you know that there's something in between. So yeah. All right. So how do we do this? We make the DNA small by shredding it. You shred it. In the old days, you used to put it. You suck it up in a syringe with a needle on the end of it, and you just go like that. So to push it through the needle a couple of times, the sheer force of running through the needle just shreds that like spaghetti out of a little kid's mouth, or really cheap noodles. It just it's a mess, and that's fine. That's what we want. We want little bits of DNA that are about the right size on average for our sequencing. Okay, once we have shreddified the DNA, then we're going to attach little bits to either end of it. So this is double-stranded DNA, and so we can attach what they call adapters to either end. And so they'll call them the A adapter and the B adapter. If you're, I don't want to say colorblind, color challenged, I don't know. I guess you can still read the letters, yeah. But A is supposed to be green, B is supposed to be red. They're not actually that color. I'm just showing you in this diagram. And so you can glue those onto the end of the DNA using an enzyme called DNA ligase. You know what? There's a whole molecular biology lesson there, but let's just assume that DNA ligase will ligate or join two pieces of DNA together. And that's a good assumption. That's an assumption you can rely on. All right, so now we've got random sequences glued to both ends. But because we're doing this on billions and billions of pieces of DNA at the same time, we're actually going to try to go fast rather than go carefully. So instead of putting everything onto the front end and then flipping it over and putting everything onto the back end, which we can't do because DNA doesn't have ends, it just has, or rather it doesn't have a front and a back that are obvious. It just has two ends that kind of look the same. So we're just going to glue these on. And so you put some A's and some B's in there. You tell DNA ligase, you know what? Put some A's and some B's on. And then afterward, we can sort. And anything that has an A at one end and a B at the other end, we're able to pull out. I think they usually use a streptavidin biotin pull down for that. But that's not important. That's just how it's done if you needed to know. If that question was burning in your brain. And so you get all these out. And so they have an A at one end and a B at the other end. That's great. That means we know what the end of each sequence is. Now, do you see where we're going with this? If we know that sequence, we can just do sequencing because we can get an oligo because we know what the sequence is, and we can bind an oligo to that, a little primer, and then just sequence. Easy, right? Yeah. No brain. Yeah. Just works. So there it is. That's one part of our little magic show. Yeah. The rabbit was, under the table, and they put it in the hat. Yeah. Sorry. It's not very interesting, is it? All right, so now we've got these sorted, and we're doing that so we have two different things, which are going to be like different primers, like a forward primer and a reverse primer, and we'll know exactly which ones because those are the ones that we installed on the ends of the DNA. We don't know what's in the middle, but since we know the ends, we'll be able to read the middle because that's how sequencing works. All right, then we're going to make some salad dressing, and by salad dressing, I mean the gross kind that's like oil and vinegar, and I hate vinegar. So yeah, if you do this stuff, I don't know, you're wrong. Yeah, stop it. Make the world a better place for once. Sheesh. Yeah. Not really, but I don't know, kind of. Let's see. Yeah, sidetrack. There we go. So we're going to do PCR, but we're going to do PCR on a little bead, and the bead is covered in oligos. Now, which oligos did we put on the bead? Can you take a moment and figure that out? I bet they're the same ones that will bind to the A sequence we just put on, or the B sequence, because who cares? Yeah. And that is actually correct. Yeah. There's some of these that'll bind the A sequence and some that'll bind the B sequence. So, all right, good. We're doing great. And all right, once we've done that, we will now have we have a lot of beads and a little bit of DNA, so that mostly the beads don't have anything on them, but if they do get something, they'll probably only get one DNA on them. Occasionally, it's going to happen, and we're going to get two different DNAs on the same bead. that's not going to get us a usable sequence. That's going to be garbage, and we'll have to throw that out at the end. But, it's okay. We'll be able to tell because it'll look like chaos. It'll look like two sequences happening at the same time, and we won't be able to tell which is which. All right, then we're going to get all those things. Do you remember from like a couple lectures ago? We listed out all the components you need to do PCR. Those are the components you need right now. So yeah, go. Get them together. Get to the kitchen. Yeah. Assemble me a nice PCR brew, and we're going to have the PCR mix and the DNA and the beads, and then we're going to put a whole bunch of oil on top, and then they shake it up, and they do what's called emulsifying it, which is just mixing oily things and watery things so that they kind of stay separate for a little while. And these are going to stay separate long enough for the PCR to actually work. So now each of these little beads are very hydrophilic, and so the water clusters around them. And in the water, we have Taq polymerase, magnesium, nucleotides, all that stuff. So in one tube, we can have millions of little beads, and each bead can have its own little piece of DNA, and each one of them can do PCR in its own little bubble of PCR mix instead of being all together. And so they might only make 50, 100, maybe 1,000 copies of one piece of DNA, but that's plenty. That's all you need to be able to sequence it in this technique. So it's kind of cool. Yeah, kind of cute too. I like the little beads in there. All right, so we're getting places now. We're starting to move. All right, so let's see. We added our water, we added our magnesium and all that other stuff. Oil and water do not mix. Yeah, there you go. You heard it here first. We just heat up the entire tube, which heats up all the little water droplets, and all of the little PCRs happen at the same time. So we've now successfully copied a bunch of things, and we end up with a whole bunch. Every time we copy one, we'll denature, and because the DNA can't go anywhere else, it'll go over and it'll bind to one of the other primers on the bead. And so we'll just keep copying that DNA right there in that little bead over and over and over again. Really cool. Yeah, very cool. Okay, then we take our beads and we get rid of all the beads that didn't have any DNA on them. They have a good way to do that. And all these beads get loaded into what looks like the world's largest honeycomb. If you have that fear of holes, made up disease from the internet, this picture will be particularly horrifying. Yeah, don't stare at it. Do yourself a favor. But they're just the right size so that one bead can fit in and not two beads. And so that's great. And so we load these little DNA-covered beads into the little wells, and then you cover everything over just sort of with a little slurry of teeny tiny beads. And these are called enzyme beads. It's just like a freeze-dried or sort of dried-out enzyme mix that has some enzymes and some chemicals, which are exactly what's going to make this technique work. All right, now we're really rolling. So each little well, each of those little cups, has one bead inside, and each bead has one kind of DNA all around it. Many, many copies. Yeah. All right. So now we just add those little enzyme beads. And the enzyme beads have a polymerase. It's Taq polymerase or something very similar to that. There's a picture of Taq polymerase again because, yeah, I like the pictures. They look nice. That's why I have this picture up here as well. Just looks nice. I think that's a hemoglobin. Yeah. I don't know. Could be something else. Who cares? Let's see. And so when we add a new nucleotide, we're going to release pyrophosphate. That's it. And so the way the technique works is you got these little beads there, and that's fine. And you're going to wash over all the beads at the same time with one nucleotide. Let's say A, the nucleotide adenosine triphosphate. Great. Awesome. Deoxyadenosine triphosphate, actually, because we're copying DNA. This technique works with DNA. And every one of those Taq polymerases will be sitting there on the end of the strand. It'll have that little primer that is part of the bead, and it'll have its template bound to that because that's how we set this up. And if there's a T sitting right there waiting to be copied, then the A is the perfect thing. It'll take the A, it'll put it in, boom, and it will add an A. And when it does, it'll release two phosphates. And you know what? If there's two T's there in a row, It'll add an A and then another A. It'll keep adding A's until it runs out of T's to add A's to. Yeah. Or to add A's that are complementary to, if that makes sense. Yeah. There's a nice video. You can watch it at the end, and it'll bring things together. But I don't know if the video works without all the background. Yeah, it's faster for sure. All right, so we add a nucleotide, and we're going to release a pyrophosphate, double phosphate. The pyrophosphate will then start this chain reaction, which is catalyzed by all the other enzymes and things in the bead. So let's look at what they are. First enzyme, ATP sulfurylase. It's going to take this adenosine phosphosulfate or something like that. I'm not sure what the APS stands for. And see how it's like, this is ATP. So there's the A part. This is the ribose. This is the side. And so looking at this, it's not deoxyribose, it's ribose. That means it's ATP, the energy source. Yeah. ATP, like what you eat your, I don't know, breakfast cereal to make, right? Yeah. Well, didn't your mom tell you you gotta make some ATP, eat your breakfast? But what we have, that's what we want. Because if we have ATP, we can use it to power a reaction. What we have is this thing, this APS, which has one phosphate and then a sulfate that's blocking the end of it. But look at that. We just put in a pyrophosphate. And so if you have a pyrophosphate and an APS molecule, and there's tons of the APS molecules in this enzyme beads, and you have an ATP sulfurylase, why, that will take this and it'll put on the two phosphates, take off the sulfate, and so you get a free sulfate, and you get an ATP with 3 phosphates on it. Boom! Voila! So if there's pyrophosphate, you're going to make an ATP with ATP sulfurylase. Yeah, sounds like an advertisement. Okay, what are we going to do with that ATP? The coolest thing, we're going to make it light up. So there's the enzyme luciferase. This is the one from fireflies. And that enzyme catalyzes one particular reaction. So every time it makes a light, it has to waste an ATP and it has to destroy a molecule called luciferin. So luciferin is this green molecule right down here, and whatever, the bugs make it, but so do our chemists, and so we load those beads with a lot of luciferin. Yeah, if only there was some ATP, we could complete this reaction. The ATP is the pink molecule. Here's the 3 phosphates up here. And so we're actually going to bind this. We're going to release the phosphate off the end of it, and we're going to make, boom, a little flash of light that was started because we have a little bit of ATP. The ATP was the only thing that was missing. And so, yeah, we get a little flash of light. How do you see a flash of light? The camera, of course. So all you do is you take a very high-res camera, you set it above this thing, and you look at the wells. So you put a nucleotide on, and you see which wells light up. And if they light up, if they put in more than one A, they'll make more pyrophosphate, and so they light up more. And so you'll see a brighter light from any of the cells where they put in 2. And it'll be about twice as bright as the light that you get for putting in 1. And you know what? If they put in 3 or 4 or 5, you can tell. It gets a little hard after like 7 or 8 to tell. Like 8 from 9 is a little difficult, but it can be done. Yeah. This technique struggles with what we call homopolymers, where it's a lot of the same nucleotide in a row. But, that's not the usual case. All right. And so then, awesome. We made a little light, and the little light tells you that we put in an A or multiple A's. And so we just have a record of, okay, what's well 1 doing? Like, okay, put in an A. Well 2 did not put in an A. Fine. And so we know well 1, the first nucleotide, is an A. Great. Yeah. Then we just have to clean up because we have all those wells where we had A and they had a C sitting there. They're waiting for a G to come along. They can't put in an A. They can't do anything with an A. And so we have an enzyme called apyrase, and it comes in and it just takes off the phosphates. It takes off the 3rd phosphate, then it takes off the 2nd phosphate, the gamma and beta phosphates, if you're counting along in chemistry language. Once it's done that, these are no longer useful. So it'll get rid of any leftover nucleotides that didn't get used first. And then once you're done with that, you know what? Now it's time for a different nucleotide. Let's do C. And so you'll run C over, and you'll see which wells light up, and then eventually that'll stop. The apyrase will work. Then you say, great, let's do G. And you run G over, and then you run T over, and you start over again. You run A past. You run C, you run G, you run T. You're just like literally flowing these things over the top of the cell. The enzymes will use the correct nucleotides if if that's the next one that goes in. And if they use that nucleotide, you will see a light coming from that cell because that's the little cup where the bead is and where the PCR is happening. That's it. That's pyrosequencing, pyrophosphate illumination luciferase based sequencing. Pretty cool, right? Yeah. And so you can have 1,000,000 of those little beads on a great big plate, great big like this big. And you can read all those sequences at the same time. And you may get 200 nucleotides out of each one of those. With just a couple of plates of that, you can actually sequence large eukaryotic genomes all in a snap. It's about as easy as it's gonna get. So there you go. And this is the link for the video. And here again, I know you know how to watch a YouTube video, so you can go watch that video. This is a little screen grab from it. But yeah, it's good. It kind of shows what we just talked about. So yeah, get that nice and solid in your brain, and then we can go on to the last level of DNA sequencing, the final boss. Yeah, coming up. Gonna be exciting.